Thank you so much, Simon. Thank you all for coming. I mean, I guess the rain didn't stop you. The food helps, I'm sure. I'm looking forward to checking that out too, but thank you for coming anyway. So before I get into navigation, I mean, I think the classic picture of the fly that many people have is it's sort of this reflex machine, right? Things happen in the environment and the fly just reacts to it, just responds to it. And so if there's one thing I'm hoping that you'll leave the talk with, it's the notion that maybe there's more going on here than just that. I mean, reflexes are an incredibly wonderful part of insect behavior. I mean, they're remarkably fast, they're robust, and so on and so forth, but insects have to do a lot more than just that, and so hopefully you'll see that over the course of the talk. So to start off with, do flies even really do much navigation? And so Michael Reiser, who's a colleague of mine at Genelia, had this collaborative study with Charles Zucker a few years ago, where they did what's the equivalent of the rodent Morris water maze task. So this is a task that I'm sure those of you who work in rats know well. A rodent is put in a pool of milky white water. It's maybe this large, maybe a bit larger. There's a hidden platform submerged under the water. It can't see the platform. You put the rodent in there. Over time, the rodent kind of figures out where the platform is, doesn't like to be in open water too much, kind of swims over to it, hangs out there. Okay? And it uses visual cues to get there faster and faster over time. So Michael Reiser was wondering if he could do the same thing in flies. And so, of course, he didn't put them in water. That doesn't work so well. But what he did do was he came up with this idea of having one cool spot in an otherwise hot floor. Okay, so there's one spot that's at a reasonable temperature. This is super hot. If you put a bunch of flies there, this is what happens. The flies run around like crazy. This is a sped up video, so they're really running around like crazy. Eventually, they find their way to this cool spot, which is going to be marked with this yellow dotted line. Most of them do anyway. Spot moves, but you'll note that the visual panorama moves with it. So the question is, will the flies get faster and faster over time to get to this target area as they start to make the connection between the presence of these landmarks and the particular spot that gives them safety? So not all of them get it, right? I mean, some of them clearly don't. But you'll notice the next move, I mean, some flies just go directly to that spot. I mean, almost immediately they're going to the right spot using those cues. And so they did a bunch of control experiments, with just the thermal cues, none of the visual cues, and the flies cannot solve the task. And they really need the visual cues in order to be able to do this. And of course, they did what's called the probe trial, where there are actually no thermal cues whatsoever. The entire floor is hot, and they just ask, where do the flies end up? Where do they go when they have no thermal cues to help them? And they tend to hang out, or they first try the locations near where the cool spot is supposed to be. So did the task make sense? Essentially, you use visual cues to get around, find your way to safety. Flies tend to be able to do that. In this task, these are not flies that have been trained over weeks. This is literally the first time they've seen this task, and in minutes, they kind of get it. So a more recent task, where it's not really a task, it's just a condition in a way that flies attain. So if flies are just put in an empty arena like this, they'll often wonder about the sides of the arena. But if, as in Michael Dickinson's experiments, Irene Kim and Michael Dickinson's experiments, you put a little droplet of food there, something that the fly grabs onto and then kind of consumes and it's gone, flies will initiate a very different sort of behavior. They do this thing called a local search. So they'll just wind around this area, leave that spot, search around, come back to that original spot and go round and round again, especially if it's a nutritive food like sugar or something. That's what they do. So it's as if they're searching, but with a home base in there. And what this gave them an opportunity to investigate was whether or not flies use what's called path integration. So the idea is, if I knew that this was a spot I needed to come back to, and let's say I didn't have visual cues, would I be able to kind of wander off like this, maybe make a turn, keep track of how far I'm going, come back, and roughly, with some security so that I don't smash into it, stop here and I'm pretty close, right? So the way I did that was I kept track of all my movements. I have sort of an internal representation of how far I'm walking, how fast I'm walking. I'm kind of able to keep track of my turns, and I can come back to the same spot even though I'm not using visual cues. Turns out flies can do that. So if that's the central spot, what Michael Dickinson's lab showed is that flies can take these trajectories, but as they go further away from that central spot, they change the angles at which they walk and the angles at which they come back so that they can come back to that original spot. And they showed this in darkness and light and so on and so forth. Even without odor cues there, the flies can kind of find their way back to that spot. And so there's a notion then that somewhere in the fly brain, clearly it must keep track of its location. Not like, not location in two-dimensional space here, but still location enough that it knows how far it's gotten away from something and which angles it took. All right? So that's something that we'd call an internal representation. The way you and I do it is using an internal representation, or so it's thought. And people have done experiments in rodents where they've recorded from these neurons called head direction cells. And so if you're trying to keep track of angles, well, it's helpful to kind of have an orientation map in your head of different angles in the space you're in. And so flies tend to do this where, not flies, I'm sorry, rodents tend to do this thing where they have these neurons in particular thalamic regions and other parts of the brain as well that are very sensitive, and they spike a lot. So they fire a lot to particular directions in heading space. So if they're pointed like this, maybe if one set of neurons is going, another one, and they're tuned based on the heading direction that the animal's in. And so there's been a ton of work, primarily theoretical, to explore how you might generate an activity pattern that's so localized in heading. And so the kinds of models people have used are so-called ring attractor models, and there's again a long history of this from the Sompolinsky group on to many, many others. Wu and Amari may be originally proposing a network of this sort, but the general idea is that if you wanted to keep track of heading and you wanted a unique representation of heading, one representation that tells you you're pointed in this direction versus this, and so on down the line, well, you could do better than, you couldn't do better than arrange neurons in what is topologically, and I state this again, topology-wise, a circle where each neuron is connected excitatorily to the neurons near it. So in other words, if I'm tuned to this direction, I lightly excite the ones that are tuned to this direction and this direction, okay? But I really inhibit the neurons that are tuned to the opposite direction because I don't want to confuse my heading representation with the one that's dramatically opposite. You could say, so essentially you have inhibition on all these units that have heading preferences in the opposite direction and excitatory coupling to the ones that are in the same direction, okay? So this is just an idea though. These are kind of computational models, beautiful models. This is called a ring attractor. But what we've been able to kind of investigate is, well, maybe the fly kind of has something like this. And this is where this story is going to go. And we're going to talk about roughly how this kind of thing works in the fly to some extent. We're starting to have insights there, us and others. And then this particular brain region is the one we're going to focus on today, which is where you find networks like this in the fly. And so this is kind of your quick introduction to the parts of the fly brain that are relevant to the talk today. So there's the visual system. Inputs from the visual system come into these central brain regions. But I should say most visual inputs go elsewhere, and many of them go directly down to the motor areas and control lots of really cool reflexive behaviors as well as behaviors that you wouldn't call reflexive but are also unfold very quickly. This region, on the other hand, it's where things rattle around a little bit. So there's recurrently connected circuits here. Things kind of unfold a little bit more slowly there. And that's the region we're going to focus on primarily, okay? So this region is called the central complex. And it is very central, and increasingly it's clear that it was well named because it's pretty complex as well. So this region is called the ellipsoid body, this circular thing here. This is called the protocerebral bridge. And I should say that there are many other subregions within the central complex. These aren't the only parts of the brain in the central complex. There are others. But for today, we're going to focus primarily on these two. This is something that you find across many phyla. So it's in lobsters. There's something called the central body. It's in flies. It's in dung beetles. It's in monarch butterflies. It's in grasshoppers and locusts. So work in a bunch of these systems suggests that this region might be involved in navigation. And when I say navigation for a monarch butterfly, we're talking about hundreds and hundreds of kilometers, so not just something in a small space. So each of these animals has a different kind of little thing that it niche that it navigates within. And in this case, it's not little at all, okay? But it's shared across these animals. And so one reason why we thought we could come in and maybe do something interesting here is because from the work of these animals, it's clear that there are sort of sensory maps in these brain regions. So there's basically a map of what's around you seems to be in there. And so we thought, well, we could come in and maybe look at exactly how those maps work. And a key technology that we had to develop in order to be able to study this properly was to put the flies in a situation where they were actually able to behave while we were recording from them. And so this was a decade ago now. But what the kinds of preparations we use are the flies on an air-supported ball. We call it a Buchner ball after Eric Buchner, who in the 70s came up with this very nice idea. And so it's an air-supported ball. The fly kind of can run on it. and then we track the movements of the fly on the ball, and we feed that back in a closed loop into a visual arena. So we can operate it in different ways, open loop or closed loop, but essentially imagine virtual reality for the fly, okay? So one mode is when it's walking, a different mode might be it's flapping its wings. So Simon does these kinds of experiments in moths here, but basically the fly is flapping its wings, and we track the wing beats, and if the fly wing beat suggests to us that the fly intends to turn in this direction, we move the world this way. Opposite direction, we move it the opposite way. And so all we control is yaw. So unlike some of you here who maybe in the moth are doing two-dimensional tracking and virtual reality, here we're doing one dimension in the fly and 2 dimensions in walking, all right? So what did we focus on? We first started to look in the ellipsoid body at a particular class of neurons. Don't let the names bother you too much. EPG just means it goes from the ellipsoid body to the protocerebral bridge. The P stands for protocerebral bridge, and another region called the GAL, but we won't talk about that yet. So what do these neurons do? Each neuron has its dendrites, primarily dendrites, in one little slice of this donut-shaped thing. So this is the ellipsoid body, and each neuron, each different color, this is a stochastically labeled sample here. So each color is a separate neuron, and they sort of tile this donut, okay? Each one is in a slice. And so what Johannes Selig in the lab did was put GCAMP, which is a calcium indicator, in these neurons, and you'll see why we call them compass neurons, because this is the population activity of this population. Each neuron, again, has arbors in just one slice of this, but they sort of organize into this little cluster, into this, what we call a bump. And so the activity is localized, and in this particular case, when the fly is walking on the ball and it's looking at this visual scene, so the visual scene, I should say, is wrapped around the fly, so it's not just a little square patch or rectangular patch in front of the fly, and you'll see this bump of activity tracking the fly's orientation in that scene. So again, the scene is moving based on the fly's movements, and hopefully you can see that the bump tracks the fly's movements, okay? So once again, basically, just to be clear, this is not just a literal representation of what's on the visual display here, right? So this is not just a retinotopic map of what's there. This really is something that represents something else, and we have good reason to believe that it is, in fact, the orientation of the fly. And one reason we know that is we can take the neural activity, use the fact that we know how these different neurons carve up this structure, divide it up into about that many little wedges, and then compute from that a population vector average. So this is just an old-fashioned kind of way of doing things. You compute the population vector average based on the activity profile in these different areas, and you come up with a net vector, and you call that kind of the population vector average representation of orientation. Well, if we take that orientation estimate and we open up this elliptical thing, this circular thing, into 1 through 16 like that. So basically you're looking at 1, 2, 3 all the way down to 16, but now we're going to track it over time. What's the activity profile over time? So you're going to see a plot where basically we're going to track this along time. If we actually watch what happens over time, what you see is that the population vector average estimate, this brown line, really nicely tracks the orientation in that panorama, okay? So basically, we could say that if the fly were using this kind of representation in order to keep track of its orientation, it would do pretty darn well, right? So now, let's put it in darkness. How does the fly do when you take away its visual cues and just give it nothing to look at, basically? Turns out the fly does just fine, or rather this structure does just fine. So this shouldn't surprise you. You already saw in the earlier slide, I told you that in the Michael Dickinson assay, flies were able to do this thing where they were able to come back to the original spot they were at in darkness. Thereby, you should assume that they're keeping track of their orientations in darkness as well. And so here's a representation then that works in light with visual cues, works in darkness as well, kind of like you and me being able to track how much we're turning, when we're going after a piñata or playing something else. Further, what was kind of remarkable was not only do they do this, if the fly just happens to be chilling out, just standing there in darkness, in pitch darkness, its eyes are black and it really can't see anything, it's just sitting there, occasionally grooming, but basically sitting there. Sometimes we have clear proof that this thing can actually persist through all that. 30 seconds, 35 seconds, just as long as the fly happened to be standing there, this representation persists. So here's where really you should think hard about, when you think about that insect in front of you that you're looking to squash. I mean, yes, it can escape that attempt to squash it, but while it's sitting there, there's a lot going on in there. I mean, it knows where it is, right? That's kind of amazing if you think of this tiny little brain creature and what it's capable of. And so for us, this has been kind of the basis of a bunch of studies, and so this is older work. But where we've kind of gone with this is to ask, how do you generate these kinds of representations? Because the challenge in other systems is not to show that such representations exist. Like I said, in the rodent, people have recorded from head direction cells. Now here you can see the complete population, which is kind of cool, but still, fundamentally, what we think the fly gives us an opportunity to do is to really get to the nitty-gritty of how you create a representation like this. And so that's what we've been doing for the last several years. And so the first little bit I'm going to tell you about is how it works in darkness. So how do you move the bump around, basically? And so the key behind that, we think, is a recurrent loop with neurons that actually come back from this structure called the protocerebral bridge. And why do I say come back? Well, thanks to Tanya Wolf's work, what we know about these neuron populations is, so there's these EPG neurons that we thought, at least at the time, take input from the ellipsoid body and then convert it into, take it to the protocerebral bridge. But there are a complementary set of neurons called the PEN neurons that we really latched on to, as did Gabby Mehman's lab at Rockefeller, and Jonathan Green in particular, his then grad student. But what we both latched on to was that there's a curiosity with these neurons that Tanya Wolf discovered, which is that these guys get their input in the same places that the EPG neurons drop it. However, they come back with a shift. Okay, so if you look at these two sides of the protocerebral bridge, imagine handlebars of a bicycle. Input from here goes to some column there. These are very stereotyped morphologically. The connections can be plastic and stuff, but the morphology is super stereotyped. The PEN neurons pick up the input there and bring it back, but with shifted to one side. If it's an EPG neuron that goes in that direction, it comes back going to the other side. Okay, so you've got this crisscrossing recurrent loop with a shift. Why would you have some?
Up there are the facets.
These are, each of these you can think of as a glomerulus, like a compartment with axons and dendrites in there mixing up. The cell bodies in the insect and the invertebrate systems in general tend to be away from all the sites of the action. So the action's kind of in these little compartments. And so here's basically a little compartment where the outputs of this set of neurons interacts with the inputs of the others, the dendrites of the others, and this guy goes back but with an offset shift. Why would you do that? Well, if you continue to imagine this as handlebars of a bicycle, if whenever the bicycle turned one way or the other, either this handle went up or that handle went up, so essentially it's like if I'm turning one way, you would do this, if I turn the other way, you do this. If that were the case, then these neurons would potentially be providing angular velocity input to this structure, and specifically maybe to these neurons. So every time, let's say this goes up, that means angular velocity input comes to this side, and if the bump were originally here, you could move the bump down this direction every time the fly turned one way. If this side went up, which meant the angular velocity was the opposite, well, then this thing would basically get input and the bump would kind of move down the other way. Does that make sense? So if you start with the bump here, if I turn one way, the bump moves down here, turn the other way, the bump moves down there. It's like a compass needle shifting around based on which direction the animal is moving. Much easier to just show you in an animation. So Emily made this animation. These are the EPG neurons. Again, they tile the entire structure, but we're going to be focusing on these central ones that go to these columns in the bridge. These are the PEN neurons getting their input from exactly those columns, but coming back with shifts to the left and right, to the clockwise and counterclockwise side. We're now going to give the EPG neurons a bump of activity, just like you saw in the videos earlier. That propagates up to these parts of the protocerebral bridge. And so you'll see a tiny tinge of blue here that represents the PEN neurons getting activated. But every time the animal turns one way, maybe this half of the bridge gets activated, provides input here, drags the compass needle down one way. If, on the other hand, the fly turns in the other direction, it would be this half getting activated. And that would provide input neighboring to the original spot of the bump. And it would keep moving. If the animal keeps turning, it keeps moving, and the bump keeps moving around. So does that make sense? So essentially, you have a compass needle that is allowed to kind of move around based on this recurrent loop with this shift in a velocity-dependent way. Does that basically make sense? Yes. All right, so how do we test this kind of conceptual scheme? So the way that we sought to test it was Stephanie bravely decided, well, let's actually patch these neurons. So record wholesale patch clamp from these neurons in the fly while it's walking. And so she took her electrodes, patched these neurons, these PEN neurons, and asked, Well, if this is true, if this model is true, we should see angular velocity coding, and we should see also coding for heading, because I'm claiming this is input to these neurons comes from EPG neurons and from some source of angular velocity, and it should be unidirectional kind of in its tuning. So when she recorded from these neurons, she found there was indeed angular velocity tuning. So she looked at, whenever the animal moved one way versus the other, and asked if there were more spikes then versus not. And she quantified this in tuning curves. And so every time she patched onto neurons from one half of the protocerebral bridge, the PEN neurons here, she got tuning curves like this. So it spikes more when the animal turns in this direction, in the counterclockwise direction. And she got tuning curves in the other direction when she patched neurons from the other half of the protocerebral bridge, basically suggesting that they are tuned to angular velocities in the clockwise direction. So you now have angular velocity tuning, but you'd have noticed that it looks like there's a lot of variability at these spots. Where is that coming from? Well, again, they receive not just angular velocity input in the scheme, but they also receive heading information. So it's a combinatorial thing, conjunctive thing that gives rise to their tuning. And so if you place this in a two-dimensional tuning map, so basically this is angular velocity, this is heading, well, they need a particular combination of both heading and angular velocity in order to fire maximally. So if it's not the right heading, they won't fire that much, which is the blue you see over here. Okay, so does this plot make sense? Essentially, you need to be in the right heading space and the right angular velocity space in order for these neurons to go. So it looks like you have a representation then that basically provides you with this model, the thing you need for this model. And so Hervé Rouault, who's a theorist, came up with a nice little model for this, which included just the EPGs and the PENs, just the same two populations we've been talking about, connected recurrently but with this offset. But in order to get a bump, a unique representation, and not have this representation be spread out, he also needed inhibition in the network, something that was from a different study that he was importantly involved in as well. And so these we thought come from a certain population. There are multiple sources of it, but one population we thought were these inhibitory neurons. You don't need to remember their name, but just they have a provocative name. So I mean, it's called delta 7s because of peculiarities we can get into at lunch or something. But basically, there's another set of neurons that we thought provided inhibition. And so with this model, basically this is a ring attractor model, he can get something that very closely mimics the kind of heading, simulated heading, looks very close to what the heading representation should actually look like when you feed it realistic velocities. So he takes the statistics of actual turns the animal makes, feeds that into this kind of model, and we can kind of recapture a good number of those statistics just like you'd expect. Further, if we were to say, well, is this PEN stuff really that important, and we kill that input, as Dan Turner Evans or Dante did, if you kill the input coming back, the recurrent loop coming back, we could show that this is a thermogenetic manipulation. So if you silence the output of these neurons at high temperature, you could show that the bump amplitude, so the amplitude of this heading representation, the compass thing, just dropped as a consequence. So it doesn't mean they're the only things that are responsible for the amplitude of this representation, but clearly it's important. So in principle, I've kind of shown you that there's this unique representation of heading in this circuit. The heading is updated in darkness using angular velocity inputs, and it persists in the absence of external input. And I've just shown you, well, this model can kind of do all those things, and the parts of those that I haven't shown you, just trust me, they can. And so in principle, one is tempted to put up a sign like this. And it's a temptation that, of course, in all situations one must resist, but in this case, it turns out as well, it's not over. And it turns out, well, there's a bunch of little predictions that these things make that haven't been verified yet, right? So one is you'd expect there to be excitatory connections from these EPG neurons up there, excitatory connections from these PEN neurons, because that's what we assumed, inhibition from these delta 7 neurons. How do we go about testing this? So we've started to really dig deep. This is the mechanistic part of this. We're starting to get to RNA sequencing of these neuron types and asking, are these neurons indeed excitatory and inhibitory in the ways that we expect? And it turns out, to some extent, yes. So these neurons do exist, these broad inhibitory neurons that could potentially help make this happen. They are inhibitory. There are cholinergic acetylcholine-based neurons, the EPGs. We're still testing the other ones, but it looks like that's not so bad. That looks consistent with the model. Are they actually structurally connected in the way that we expect? Well, for that, we go to electron microscopy. And so we're asking there, well, if you look at neurons that go from the ellipsoid body to the protocerebral bridge, you can sort of see these structures here. They're not actually blue, right? I mean, this is just my coloring for you to see it easier. This is what it looks like in electron microscopy world. But this is a collaboration with Davi Bock, his lab, and a whole bunch of undergrads who've been tracing neurons and so on and so forth. And so what we saw there was, well, in this stack, if we were to trace neurons, so this is the entire stack, well, not the entire stack, slices from that stack, and we can literally manually go in a software called CatMade that Stefan Saalfeld, Albert Cardona, and others developed. We can basically go in and trace out, these are input neurons that I'll come to later called ring neurons, these blue ones, but the purple ones are the neurons we've been talking about, the compass neurons. And I'm just showing you one little piece. This is the ellipsoid body. I'm just showing you one piece. And they go up to the protocerebral bridge, talk to the red neurons, which are the PEN neurons, come back. So there are synapses. We can count the synapses. We can map it down to the nitty-gritty level and ask, are these neurons really connected in the way that we expect? And so it turns out the delta 7s really do talk to these neurons, the PNs and the EPGs do talk mutually and so on. And so in principle, we can verify some of these connections. So are these connections there? Well, it turns out yes. So the EPGs do talk to the PNs in this protocerebral bridge. The ellipsoid body, there are connections between the PNs and the EPGs. The delta 7s do indeed inhibit, actually they inhibit both the EPGs and the PNs. All looks well with the world. And then there are these minor inconveniences. There's a whole bunch of minor inconveniences. I'm just going to tell you one. One of which is that these things aren't really pure input and output the way I've told you. It's actually that there's back and forth chatter between these neurons even within this structure. You'd say, well, why does that matter? I mean, it does matter, right? Because in some sense, the recurrent loop isn't really needed in order to keep this activity going. There may be chatter within these things. So the compass, the bump of activity may persist partly because of interactions even within this. This has consequences for how stable the whole thing is, how mobile this bump is going to be when you push it, right? Because if there's strong back and forth activity that keeps it there, well, how do you get the compass needle to move? And so we're trying to explore the drift that you might have. So what do I mean by drift? So if I'm actually, I start to drift out of view of the Emory folks, I'm going to push myself back here. So if I were to just keep track of my orientation or just, my heading by just, I shut my eyes and I just keep track of it, my representation, is it going to drift away? And am I going to be wrong essentially in my estimate if you were to ask me a few seconds later? Well, that's sort of the problem the fly needs to solve here. If that bump drifts away, it's essentially an incorrect estimate of its orientation. And so we're trying to figure out, do these kinds of connections, these back and forth connections, potentially help stabilize the bump in a particular place when the fly does not turn? So these are things that are ongoing. I wouldn't say that we've by any chance finished anything there. So I'm going to actually say, so this is an ongoing thing that's still going, but I'm going to kind of close this part of the story and quickly tell you about another. So I don't know how far I'll get through this, but we'll see. So far I've told you about flies, the compass in darkness, but now we're going to say what happens in different visual scenes. So I just entered this room a little while ago. I already have a pretty clear sense of my heading in this room. If you're outside, you have a pretty clear sense of your heading there as well, but you're using different cues in order to get your heading in outside than inside, right? How do you quickly map essentially a scene onto some representation of heading? How do you map a scene onto a bump? And it turns out the flies will actually manage just fine in this scene and that scene because we've tried it. So we actually put these flies in virtual reality. It's a much more spectrally impoverished situation we put them in, but nonetheless, the flies do develop a stable bump that tracks their heading in this scene, which is a pretty scene from Genelia, and this scene, which is also from Genelia, but is in the woods nearby, okay? And so if you can develop a stable representation in two different visual environments, or in a ton of visual environments, clearly, somehow, you're able to kind of make the system plastic. So how do you take a visual scene and map it to a bump? That's what we wanted to ask. We already know that if from our rig experiments from before, that different flies even will have a different relationship of their scenes to the bump, okay? So this guy has his bump, or this gal in this case, because we do most of our experiments on female flies, the bump is over here relative to the visual scene, exact same visual scene, and this other fly might have a bump over there, okay? In fact, if you look across different flies, the offset of this bump relative to the visual scene is different for different flies, okay? So in other words, there's some flexibility there, and the flexibility is such that individual flies tend to be pretty stable in these offsets, like so, like so, like so. But sometimes, every now and then, we saw evidence that individual flies would also show signs of plasticity in this particular mapping between the scene and where the compass representation forms, right? So this is an arbitrary reference, and then you're kind of moving after that arbitrary reference, but that's an arbitrary thing for real, even within a single fly. So, yeah?
So different animals have different mappings of different parts of the visual scene, to different parts of the ring.
Yep.
Is there a parallel remapping of the outputs of the ring
That is our assumption. So the question is there a parallel remapping of the motor side? And we don't know that yet. This is one of the things we're actively investigating, but it's a totally relevant question. If it's arbitrary here, is there a relationship? We'd assume so, we don't know yet. So for the first part of the thing, the visual part, where does the visual input even come in? Well, there's particular visual pathways coming into these structures, into the central complex from regions like the anterior optic tubercle, the bulb. The names don't matter. The point is there's visual pathways coming in there. And multiple people have investigated it, not just from my lab and my group, but from other labs as well. But in this particular case, Johannes sort of started looking at their visual inputs in pretty fine detail. And more recently, Yi-Sun, in collaboration with a wonderful theorist colleague of mine at Janelia, Anne Hermansted's lab, they've sort of looked even more deeply at it. And what it turns out to be is if you throw on white noise stimuli and just flash these little dots at the fly, what you realize is that flies, this particular set of neurons in the bulb, have a particular kind of feature detector-like response profile. So in other words, they respond to light and dark in particular patterns in different places. Different neurons have different places that they respond to. Further, there's complex relationships for those receptive fields. They're affected by things, the other things in the environment, not just the thing in the receptive field. And they're affected by when things are presented. So if you present, for example, a stimulus now like this, and then another stimulus there, you'll get a different response than if you show them both simultaneously, okay? So there's all these complexities to the visual input, but at a vanilla level, you can assume that basically in this region, the inputs that the fly is getting is just for now, just assume a simplification, is literally like the position of something determines whether the neuron goes or not. And so you're seeing different glomeruli again. So these are little regions where dendrites and axons interact. But this is a population of neurons, and these guys are quiet now, but as this dot gets lower, you'll see these guys get activated. So different neurons basically have different receptive field properties. Again, they're more complex than this, but just for simplicity, think of this as a mapping of the visual scene onto a population of neurons. Not yet onto the bump, right? Just a population of neurons that captures visual features in the fly's surroundings. So we think that's the input coming in, and you get different flies have a bunch of recognizable feature detectors that you can see in fly after fly. So where does this go? Well, we know from electron microscopy, these are the neurons you sort of saw earlier. We've traced them, and they talk directly to the compass neurons. And so Sung-Soo Kim was basically interested in seeing, well, that communication, is that where the flexibility might lie potentially? And is this how visual scenes get mapped onto a compass representation? So just to clarify, we're talking about each of these neurons sort of specializing for features in different parts of the visual surroundings. And these purple neurons, we're saying, just give you one bump, right? The population maps to one bump. So somehow this scene is transformed into an abstract representation of my heading in this space. And so this idea has been suggested before. Bruce McNaughton's lab, again in theory work, in this case conceptual work, suggested visual inputs might come in to a bunch of neurons that would carry sort of that would carry heading information. And then they also suggested that there might be velocity neurons that move the bump around in darkness. So there's kind of ideas like this around. And more recently as well, Surya Ganguly's lab, in work that's parallel to our own, suggested that there might be plasticity in synapses. Again, they were talking about grid cells, but it's still, the general concept was the same. Ultimately, you want to be able to adjust things, adjust this mapping so that when I turn this much, the angular velocity part of this system is mapped well to the visual part of the system, so that essentially the bump moves to the same place, determined both by my angular velocity as well as by the visual scene movement. Yeah, that makes sense? That's how you kind of have a consistent representation of the world in your head. And so this is what Sung-Soo sought to look at. And so the general idea was pretty simple. It was like, well, let's put the fly in a simple world. He's done it with more complex worlds as well, where there's just a single stripe. And now let's say this is the EPG population and the bump is over there right now, because the bump, as we know, can survive in darkness as well. But now there's a visual scene and maybe some particular ring neuron gets activated. The ring neurons are these neurons that go everywhere. So this neuron gets activated. And so the idea is, well, maybe because of the co-activation of these, you can use Hebbian rules and say some association forms between the activity of these neurons and that one neuron, okay? Now, let's say that you move the scene and you say this bar is now over here, a different ring neuron might get activated. It's actually all populations, but we'll just go with the simple framing for now. And the bump moves to a different position in this ellipsoid body. And so you'd say, well, okay, maybe the association between these two, there's some Hebbian rule in operation there, and maybe you create an association there. And so over time, as the fly explores the scene, presumably you can map the world through this ring neuron's eyes, ring neuron population's eyes, onto a heading representation, okay? And so that was the general idea. And so we thought, well, can we test this directly, causally? And so the way we sought to test it is we said, well, if we can optogenetically induce a bump of activity in this part of this ellipsoid body, can we then create an association between this part now and this set of ring neurons? So essentially, can we arbitrarily create an association between a visual scene and a heading representation? Okay? And so what I'm going to show you is his experiments to do that, but first you need to know how he does that. So he uses an optogenetic reagent called CS crimson. It's a channel rhodopsin variant. And so what he does is, while doing 2-photon imaging, he's basically stimulating a small set a little bit harder. So he's running the 2-photon laser past the whole thing imaging, but he's stimulating one little piece a little bit harder. And what that effect is this is the bump originally, but it moves there because he's optogenetically smashed it there. And there's only one bump in the system. So as soon as he creates a new bump, the old bump just dies. Okay? So essentially, what you're seeing is when it's red is when he stimulates, and then the bump sort of survives and is happy thereafter. So I'll show it to you one more time. In this case, the fly is flying, but it doesn't matter in this case. Yeah. So new bump created, and it turns out mutual inhibition is what keeps it there, but that's okay. We'll leave that aside for now. So now here's the experiment. Initially, there's a certain mapping, which is in this case, this is where the visual bar is. Again, the visual bar is moving all around the fly, and it's mapped to this spot in the fly brain in the ellipsoid body. What Sang-Soo is going to do, so this is the mapping right now. This is the compass needle. This is where the Q is. Sang-Soo is going to create a new different mapping. And so here, he's optogenetically stimulating here to create an artificial bump. And this is where the visual cue is, and you'll see that the tuning, I mean, the relationship is now different. But he's going to hard code it in because he can specify where the bump needs to be, and he can hard code in as well. This is open loop now. He's putting the visual stimulus in different positions and hard coding in a new association potentially. And this is the relationship now. It's a 90-degree separation. And so if he now tests it back in closed loop, again, the fly now controls things, and he looks at what this system is. Okay, I'm running short on time a little bit, so I'll go through this a little fast. What he sees is that he can shift the bump from its original thing on this side of the visual bar to anti-clockwise, counterclockwise. He can then shift it back. So again, he's now trying to change it in the opposite direction. You get the basic training protocol. You're showing visual stimuli and smashing the bump into one particular place. And now when he tests it later, it's again 90 degrees the other direction. So basically there a little bit more than 90. So again, it started out here. He was able to move it, by forcing it to 90 degrees. He ended up getting about 80 or so. And then he goes in the other direction, moves it maybe 110. If he does this experiment after experiment, this is what the data looks like. So this is how much he attempted to shift the bump, this offset from 0 to minus 360 plus 360. And this is where it's actually moved. The diagonal tells you he's doing pretty well. This is in contrast to where if it has no optogenetic reagent and he just uses the exact same protocol of heating the fly, etc., this is what he gets. Okay, so basically you get nearly a flat line no matter what you attempt to shift it at. So you get the basic idea. Essentially, we can shift the bump, which means there is plasticity in this system, and the general idea seems to be, these are inhibitory neurons, so we think actually that they weaken their connections every time they're coactive, and what that gives rise to with a simple learning rule, kind of a Hebbian, inhibitory Hebbian rule, you can kind of get this. But in this case that I showed you, we kind of hammer the system, right? We use optogenetics, we really drive it hard. Can you see this even in natural behavior? Well, so you can ask, well, if I put the fly in this world, so right now there's a single stripe in this world, there's a bump tracking it, you'll agree that the association is kind of one-to-one. It's really locked on right now. What if you expose the fly to a world with two bars that are ambiguous? So it's basically like, there's a bar there, there's a bar there, and the bump is really nicely locked. You'd think to this, based on this information, you'd say the locking is one-to-one, except, well, at the same time, you're potentially training on the other one too, right? If the training principles I just gave you are true, then exposure to a scene where there's a bump in exactly the opposite thing should kind of also encode something. And so we can ask, did a ton of exposure to this, like many minutes of exposure to this, in this case actually just 3 or 4 minutes, does it actually make it more likely that the bump actually hops to the other place, the new offset? And indeed it does, so as you can see it here. It doesn't happen all the time. It's like a fraction of flies that you see this for the amount of time we expose it. But every now and then what you get is essentially a remapping based purely on visual experience. Okay, so it starts in a place, but then exposure to an environment, in this case an ambiguous one, basically creates another mapping too. Yeah?
Is there any destabilization of the animal's navigation ability while it's
So in this particular case, we expect it to be confused. We haven't done behavioral tests that show that, to demonstrate that it's confused, but we are doing experiments. Ask me about it later, because there are experiments where we're doing learning in the fly, and that gives us a clear goal that the fly has to go to, and then we can ask, is it more or less able to achieve that goal in the absence of the kind of compass thing that we have? Okay, so essentially then, what I've kind of shown you is our attempts to go from behaviors that we suggest would mean that the fly has internal representations of where it is in its surroundings. It's able to take multisensory input potentially associated with this internal representation. Learning, etc., is where we are hoping to go. But the end result we're hoping to get to is a mechanistic explanation. Like, take these models of how these things might be done and ask if they really are done like that in the fly, or if so, in what precise ways are they done, which allows us to examine properties of the system, the stability, and so on. Towards the 2D thing, we've kind of started making some progress in Hannah Haberken, a former grad student and now for a short while a postdoc in the lab, luckily for me, has been doing these experiments where she can optogenetically train these animals. So the brightening red was basically we heat the animals as they go close to one particular kind of landmark in their surroundings, and we cool them when they go to another, except the heating and cooling is done optogenetically. So it's their heat receptors. So they're not actually being heated, but they feel the heat, right? And so essentially she can do these experiments and train flies, and I'll skip over that so that I can save time for Q&A. She can train flies on these protocols where she starts them off just to get their naive preferences between, let's say, a triangular conical shaped landmark and a cylindrical one, trains them for 20 minutes in an environment where the cylinders are punished, and then asks, so clearly during the training protocol, you see that they avoid the cylindrical parts of the world. So there's a periodic world with many of these kinds of patterns, and they'll kind of go selectively to the conical objects. And the question is, does it retain those preferences that it develops over time after the training? And so you can see the switch here. So there's a marked preference for cylinders over cones that switches to a reasonable preference for cones over cylinders after the training. So this is kind of the difference in visits between the cones and the cylinders. So where we're going then is something where we can hopefully link these representations. We're also looking for more representations than just angular representations. We think there's representations also for how far you're walking, how fast you're walking, etc. And so we're trying to link that now to 2D navigation, learn navigation, and that's kind of the long-term thing in the lab. And so when I say the lab, these are the key people that I should particularly point you to. Johannes Selig, who is now a group leader, a Max Planck group leader at Caesar in Bonn, did the basic experiments to show that the compass was even there. Stephanie Wegner did the electrophysiology experiments that were pretty challenging. Hannah did those 2D VR experiments. Sung-Soo did the optogenetics-driven plasticity experiments, as well as the one showing mutual inhibition. A whole bunch of other people I need to thank, but I'll particularly point out Tanya Wolf, who's a fabulous anatomist, doing tons of really beautiful work that drives a lot of labs, not just mine. And we collaborate a lot with Anne Hermanstead, a theorist in the lab, not in the lab, I'm sorry, who has her own lab. In addition, we benefit from a ton of advice and discussions from others at Janelia. So the fellowship extends out quite a bit to many of my colleagues, Davi Bok and Michael Reiser, I'll mention in particular, but we benefit from a whole bunch of others as well. And so with that, I'll just close with a couple of statements. So when Simon introduced me, he stumbled through the mechanistic cognitive neuroscience, which many people kind of look at me like, what the heck is that anyway? And so I'd be happy to talk about it later if you like, but essentially it's a program where we're planning to use different systems, right now, many of you know, we already have fish at Janelia and rodents and flies. We're going to start with those systems, maybe add others. But the general idea is to find unifying themes and principles for higher level behavior, internal representations, recurrent network dynamics, that maybe we can use theory to unify some of these different circuit motifs in different systems. Maybe you identify the same circuit motifs, but they work differently in different systems. So we will see, but it's basically a theory experiment collaboration that maybe many of you here enjoy as well, but to specifically tackle questions that are a bit beyond, like in the higher level cognitive domain. I would encourage you to come visit. Some of you do visit Janelia, which is great, but please do come visit. There's lots of different things that will allow you to visit, so please check them out and see us there. Right? Thank you. Yeah.
So I noticed in your different visualizations of the bump that, at least in my eyes, looked like there was some variation in the amount of noise you saw around the bump.
Yep.
Is there anything through your experience that demonstrates that amount of uncertainty and where the compass points to?
It's a really good question. We don't know. It's a good question, and it's one that we're curious about. So basically, does the amplitude of the bump and the fuzziness of the bump represent something related to the uncertainty that the fly has about where it is? That's kind of the thing you're asking, right? And so it is something we wondered. We know that that region of the fly brain, the ellipsoid body, also receives input from all kinds of other things, like state, like the state of attentiveness of the fly. I don't mean attention per se, but basically something where how aroused is the fly, how awake is it? We know it gets sleep input, we know it gets satiety information. So it's entirely possible that there's a certain modulation of this whole system based on state, behavioral state, and internal state. So we haven't explored that, and that might well impact how certain the fly is, how much it cares even about knowing where it is. Yeah, Simon?
How much exposure does all of this require? And is there a mechanism for example, like when I walk in a room, I can frequently map it where, I can glance and then close my eyes and develop.
20 seconds. In a new environment, maybe sometimes it takes a minute or something. If you're switching it from a strongly encoded one to another, maybe it could take a minute, 2 minutes sometimes, but it's often really quick. Like it just maps immediately, not immediately, but very quickly. And so that raises questions of, if a fly can map a scene onto a bump in tens of seconds, what are the plasticity rules in operation? What is the mechanism for that? And we don't know yet, but we assume it's not something that requires great structural changes because it's supposed to be flexible. But we don't know what it is.
So does it take longer if they've been in the dark, or does it take
Oh.
less time if they've been in the dark?
I mean, even if they've been in the dark, there's already a pre-existing relationship there, but you'd expect it to die. We haven't explicitly looked at that, whether it's longer after the darkness, period of darkness. We don't know. Yeah, questions back there?
Following Simon, in the kind of hard hitting optogenetics that you did find still shots and animation, how much time between those shots?
That was instant. I mean, basically it's 3 minutes, 3 minutes, 3 minutes, 3 minutes. So it's literally 3 minutes of all. That's enough. 3 minutes is definitely enough, and for the optogenetics, that's certainly enough. But even in natural settings, I mean, if you just look at two different scenes, like I showed you those two different scenes from Genelia. So essentially, if you take those two scenes, it takes a couple of minutes, and then it's there. So, more questions? Yeah.
These kind of ambiguous pairing experiments are really interesting. I missed, do you have to do those in open loop, or can you do those in closed loop?
No, so we do them in closed loop because, so that was the attempt, was to say, in this ambiguous setting, if we just let the fly do its thing, how ambiguous is it in terms of the representation? And so it's closed loop, the fly is just determining where it goes in that. The assumption would be if it jumps in this way, I mean, we've seen that even in walking experiments where the fly is walking in an environment where there's only two cues and they're basically ambiguous. There's one there and one behind, and it's exactly symmetric. Every now and then the bump does jump and kind of switch. So it feels like something where if you're in a room and there's only two cues, and after a while you lose your sense of where you are and you kind of confuse being pointed in one direction with the other. We think this happens naturally to us as well, we would guess, but certainly for the fly, the representation does switch naturally with no optogenetics, no open loop, just straight closed loop. More questions?
All right, let's thank the guy.
Thank you.